All right. Hey, Zining.
Hey, Jesse.
How are you?
Good, good.
So Katie was afraid because she couldn't speak, but it looks like they needed to promote that. So
Hi, JC. Hi, Jining. Hi, Katie.
there
Hi.
she goes. Hey, how are you? Long time no seen.
Yeah.
How's everything? Where are you now, CDX?
In Los Angeles. So I moved
In
here
Los
in
Angeles.
March.
Yeah, that's what I thought. Yeah, I wasn't sure if it was in
I'm
Los
representing.
Angeles or in Oregon somewhere. So that means that you are what? 6:30 in the morning.
Yeah, it should be 6.
You are a brave man.
No, he's on mute. See ya,
Oh.
you're on the mute.
Dr. Hodley, are you awake? Did you have enough coffee?
I got a whole mug full.
Ah, there you go. There you go. Well, I am out of my quarantine, not because I got my second test, but because it has been 14 days, so damn it, you know, I did the whole quarantine thing.
There's
I
no
got
quarantine requirement in Switzerland.
Not when you get there. Switzerland is a mess because it's similar to here, you know, every canton does whatever they want. And so where I was, which is Valais, everything was closed. Restaurants were closed. Basically, what they were trying to do is lower the numbers so they could have a winter season, which probably they're not going to have. But then there were other cantons that everything was open. It's, it's very, very weird. I stayed away from people, use mask and all of that, but you never know. So my first test was negative, and the second one the results should have come yesterday, but it's taking longer. And so Anna decided today that 13 days or full room service and maid service was enough. And since the CDC says 10 days or 14 is enough, I'm done. Like I have no symptoms or anything. So obviously.
Right,
And the
guys,
first one being,
Hello, good evening, everybody. Can you hear me? This
Yes,
is
Yeah.
Tisha.
It
Yes,
seems that
yes,
the
we can
session
hear you.
is starting now, so we're live on
Okay.
YouTube.
We are live now.
Okay, so we should stop bad mouthing the
I think
ICER
you
in
already
Pune.
heard it. It's okay.
Okay, good.
Okay, good evening. Actually, good evening, good morning, good afternoon,
Good morning,
everybody.
yeah.
We have, it's been an amazing day. It's action-packed, well, more like knowledge and information-packed. We have had you all, the faculty members from across the globe, not just the faculty members, but also participants from across the globe on this virtual edition of the second TCGA workshop and conference in India. We thank you all so much. It's been a great start to this workshop and conference, and we're very happy with the way it went. So again, as many gratitudes that we can give you. Thank you so much to all of you. We do have today Dr. Sabrinathan Radhakrishnan, who will be our primary moderator for the day. He will be joining us along with the faculty members. So welcome to you all. Before we get started, I'm just going to let you know how this entire session is going to go. It is a live session. We have everybody here. We have the participants as well. Just the thing is, the questions that we have right now have been collated over the day from the various sessions that we had, 10 in total. We've collated them, and we will be, Dr. Radhakrishnan will be taking them up with the respective faculty members. So there will be some live questions also coming in. We will take them up if time permits. So we are keeping that flexible. So I hope everybody is okay with that. Yes?
Yep.
Yes.
All right. So, Dr. Radhakrishnan? He will start. In the meantime, we will probably, it's okay. We will start with the questions. The first session was on,
Okay, we're not
okay, so
hearing
the
you.
first session is in the history. We had history and milestones, data structure, including clinical information and analysis pipeline. So I'm just getting started. I'm sorry, I know Dr. Radhakrishnan would start, but since I think it's online, technical issues are expected. I'll get started. These questions will be directed to Dr. Zenklusen and Dr. Zinging Wang. The first question we have is, well, the participant says it was nice to see most of the data publicly. However, some of them are still in controlled access. For some users experience that the control data access is granted only after 6 months. Can't we get that in a short period of time?
So the reason the data is, some data is control access is because it could lead to the identification of the donors. So we make as much of the data open access as possible, but the sequencing data, cannot be de-identified. And so the raw sequences are controlled access. And in order to get access to it, you have to go through the DBGAP system. That's the way we give control access permissions. And it shouldn't take 6 months, okay? It may take a month, it may take a month and a half, maybe 2. If you complete the forms totally, when you request the access, it should be very fast. We have the data access committee that looks into these requests, and they are very, very expeditious. The problem sometimes is that people do things like, what is your role? PI. Well, the PI is not a role, okay? So you need to say, I'm an associate professor, an assistant professor, whatever you are. And then, people just, you have to have a statement of research, and the statement of research cannot be, I want to see the data. You have to say, what are you going to do with the data? Because, and that's normally why it takes 6 months, is because people don't complete the form thoroughly. And then, the data access committee says, well, what is this? And returns it to you. And so if you complete the form fully, I can guarantee that you're going to have it in a couple of weeks, 3 weeks, a month at the most.
Okay.
Yeah.
All right, that's great to know. I'll just take up one more question, and then I'm going to hand it over to Dr. Radhakrishnan. Is that okay, Dr. Radhakrishnan?
Yeah, please.
So we have a question on what fraction of the data is controlled. As a user, what can one do to access it faster?
Well, as I said, the only thing that is controlled access is the raw sequencing data, which the vast majority of users, the biologists, don't need, because really you wouldn't know what to do with it. And so, the open access data and really the access through the GDC is the best option for the biologists. If you're a bioinformatician and you want to use the raw data to check your tools or whatever, well, again, you need to go through DBGAP. You need to do the full form, fill it to the best of your ability. No, don't skimp on details, and then you're going to get access.
Okay, great. Thank you so much. And Dr. Radhakrishnan, first of all, welcome. And I'm going to let you take over the questions from here. Thank you.
Yeah, thank you, Disha. Hi, Jesse.
Hey, how are you? Good to see you.
Yeah, thank you. I just have a follow-up question on that. First one is, how often the data access committee meet so that one can expect like when they submit the application?
They actually don't meet. When the applications get to the system, they get distributed, and not the whole data access committee looks at it. And so they get distributed to a couple of members. Is rotating because we get a lot of applications, and so otherwise, it would be impossible. So it is kind of in a rotating basis. And in fact, there are certain applications that get approved by the data access committee chair because they are considered administrative, and so they don't need review. So it all depends on what your application says. And but they get submitted on basically. One of the persons in my office, Roy Tarnotzer, is part of the DAC, and he gets tasked to review, I'll say, once a week. So we are doing this on a regular, rotating, real-time basis. We don't want to have people be waiting for the access if everything is correct.
Yeah, no, that's great. Meeting once a week is already a big progress.
I think Katie has some.
Yeah, please.
I just did a renewal of mine, and once it left my institution signing officer, I think it took about 2 days to get renewed. So things seem to are pretty quick. I think it's just, that initial application and making sure you have the right information. But other than that, we don't want to hide the data. The data is there for people to use, but we want to make sure that people are using it respectfully and correctly for things that are identifiable. But all the rest of the data that's not identifiable is public and open access, gene expression, methylation, and I think the process has really been streamlined.
Yeah, that's great.
And also, I saw people always asking is, what is the percentage of data open versus closed? That question is a tricky question because of what parameter. If we are talking about data size, maybe 90% is control because the raw data sequence is so big. But in terms of number of files, 90% is open because one BAM file, we have 5 VCFs, 5 maps. So you see one file, we generate 9 files, 8 files. So majority file actually is open. And as Jesse just said, majority of users do not use control access data. And also just as Katie and Jesse said, it's like get approval, it's like 2 processes. We call it passport and visa concept. It takes you a month, but if you take you 4 weeks to get the passport, which is not really my point, our problem is give you the visa. So your institute, if it takes several months for your institute to get everything ready, that does not count. It's not on our side. We can help.
And one thing that I would say, sometimes this is set up for the U.S. system, and so you have to have a signing official, and you have to have an IT official. And we have had cases that they are small hospitals, they don't have an IT person. So you need to tell that. You need to tell that in the notes, saying, I know that you want our ITO to be signing, but we don't have an ITO because we're a small hospital. We had an issue with somebody that I actually know from Italy, and he sent me, it's driving me crazy. They keep asking about the ITO, and I go, well, explain why you don't have an ITO, and sure enough, he explained and said, we are a very small hospital. We don't have an IT officer. The signing officer, who was the director of the hospital, is the one that vouches for all the systems. And 2 days later, they have access.
I see. Yeah, I think it's clear. That's the initial process where we have to put in all the required details, and that will make the process easier. Yeah.
Yep.
Okay. Thank you. So again, this is probably the last question related to the control access. So regarding the cloud access, because like from here, downloading the data would be too heavy. And if one has to get access,
it will be probably 4 to 5 months to download all the data.
Yeah. No one is willing to try that. But in case if we have to get access, the provision to get access to cloud is the same as we have to do the dbGaP process.
Exactly. Yes. You need to have, if you want to access in the cloud. So we have a copy of all of our data, all the TCGA data in the AWS, isn't it, Z?
Both AWS and Google. So like
now we
Google,
have both AWS and Google. So good. So the thing is that data, if it's open access, again, it's open access. If it's controlled access, you need to have dbGaP access. So that applies both to downloading from the GDC or accessing in the cloud. If it's controlled, you need permission to get to that.
Okay, thanks. So, and the next question would be for you and Zhenning both. Zhenning, in your talk, you have presented something about the single cell data. Like, is TCGA planning to have some single cell data, or it's mostly the data from outside that will be explored here?
So in my presentation, I said GDC in few months we have like single cell RNA visualization tool on flight, like you can do online single cell RNA data analysis. We already have a single cell RNA process and pipeline from a software standpoint. And the actual data are actually, and Dr. Anna Lobis is going to present already, CP Tech actually already have single cell RNA-seq data in GDC, but it will be released in a few months. And Jesse can add to the TCGA is also working on that. I will leave that to Jesse.
So this is hot off the presses. We are starting a pilot program to do single cell sequencing on the old TCGA cases. We just started it. The whole idea is in the pilot to do 100 of them. Samples have been shipped to the centers, and we're going to do not only single cell RNA, which is what everybody's doing. We also are doing single cell DNA for copy number mostly, but also for mutational purposes. And we already are doing single cell ATAC-seq. So we're going to have a lot of single cell data coming out. Don't hold your breath, you turn purple, okay? It will probably be, I don't know, a year and a half until the data sees the light of day. But it's a pilot, and we have compromise from NCI that, if it works well, they will fund us to do it in as many cases as we can. So this is also one of the reasons people ask me all the time, can I get some TCGA samples? And the answer is no, because legal reasons. We have legal agreements with the sample providers that say that we're going to use it for TCGA and TCGA only. But the other reason is because these samples are very valuable, and as new technologies come along, we are using them in those technologies. And so that's why we keep whatever we have. And some samples have been totally and absolutely exhausted, so you're not going to see any more data on those. But whatever we have in the others, we're keeping them to do new and interesting data types that will see the light of the day eventually.
Yeah, sounds good. I just have one follow-up question from last year. You have mentioned about the extra whole genome sequencing going to be done on TCGA cohort.
We are doing that slowly and painfully. About, I think, 500 cases are going to see the light of day very, very soon. If not in the next GDC data release, in the one after that. And so as we are cleaning up the data and calling it, whole genome sequence ATX takes a long time to align and takes a long time to come. And so, we need to do that correctly. But the whole genomes are coming, and they are going to show up, I'll say early 2021.
Okay.
And then we'll keep adding to them. Basically, I think the first block is about 500 cases. If I don't remember, are the ovarian ones. But then, we'll keep adding one whole project at a time. The whole idea is not to give you, one whole genome here, one whole genome there, is to give it by project, so people can actually work on them. And so that's the way it's going. And we keep asking for more money from NCI to do more sequencing, and the answer seems to be always yes. So we'll keep doing it.
Okay, it's great and encouraging. Okay, so moving to the next speaker, Dr. Anna Robles. Yeah, and thanks for your presentation and a nice overflow of how to prepare for proteogenomics, the data generation and analysis. So the broad question we have is the new consortia that is starting like ICGA, how one should prepare in such a way so that they could efficiently use the tumor tissue, whatever we get, but generate multiple data types. For example, genomics, proteomics, and RNA-seq.
So I think you're referring to the International Proteogenome Consortium, ICPC?
No, I'm asking like if one has to start from scratch, for example, in the Indian context, if you have to start to do the multiomics, how one should efficiently use that.
I understand, Claude.
So it's the Indian Cancer Genomics project that they are starting. They're thinking on doing proteomics. So I think the question is, how do you make sure that your samples are good enough and your processes are good enough to profit from the opportunity?
So yeah, I understand. So optimally, you would want to proceed with a prospective collection once you have decided the cancer type that you're going to pursue. That's not always possible, but like I said, if you are interested in generating multiple data types that include post-translational modifications, you would want to, at all possible, to include a prospective collection of samples that is well controlled for the ischemia time, particularly, that means the time after resection, how soon those samples are frozen after resection, because that greatly affects your ability to look at post-translational modifications. And that's one of the most interesting data points that one can get, referring to activation of signal transduction pathways. So that's really one of the best type of information that you get out of proteomics beyond just looking at comprehensive proteome coverage. So I would say that it's one of the most important items, unless you can concentrate on a collection that has been done, let's say, in just one center, where you have some idea of the quality of that collection, in the sense that you can say with some certainty that the pre-analytical variables are controlled to a certain extent, like 30 minutes to 1 hour of ischemia time. In that sense, you could just use samples that were perhaps collected for another reason, but that have a controlled handling time and process. So does that answer the question?
Yeah. So it sounds like we have to be very quick and prepare the samples suitable.
Right. So it's all in the question of what you want to get out of that analysis. Of course, there's a lot of rich data that can come from retrospective collections, and the program did show that by using samples that were previously sequenced by TCGA, you can still add a lot of information and a lot of biology. But if you want to build a program around comprehensive characterization, then you would want to include that as one of the items that are right up at the top of what you need to think about.
Okay, yeah, that's very clear. So moving to the next question, this is switching to CPTAC, and the users are interested to know if the raw data is publicly available. And the follow-up question is there any way that we can, by using online mode, compare the mRNA and the protein data from TCGA?
Yes. So as I mentioned in my presentation, the proteomic data is being displayed as the Proteomic Data Commons, or PDC. And there, one can have access to raw data files from proteomics. Now, this data is different from sequencing, and it needs, the users need to understand how to work with proteomic data, essentially, before you can actually get information out of it. Although within the PDC, there is a feature where you can do some analysis without downloading the data. And in the future, there's likely to be more. So you can look at some of your favorite genes, so to speak. The genomic data is displayed at the GDC. And at this point, if you wanted to have both genomic and proteomic data for a single sample, you would have to go to both places and download the proteomic and the genomic data separately. The RNA data is not controlled, so you could download the expression files without having any specific permissions. And the proteomic data is completely open because that's not considered to contain identifiable information. So those are the two pieces of data that you would need. You would have to, of course, bind them by the case number, but all of that information should be self-evident from both sides. And then you would have all of the metadata associated with it, both in the some amount on the GDC and then more at the PDC. And then in order to put that together, the consortium has also developed software tools, and that also was part of my presentation. And some of those software tools are listed at the CPTAC's website. That was also listed in my presentation, and we further have training webinars on our website that show how to do this. Although probably the easiest way for a user that's starting to work with this data would be to look at some of our papers, the flagship papers, to understand how the data is being looked at to do those kinds of analysis, like correlation of protein and mRNA data at the single gene level.
Okay, yeah, thank you.
One thing though, and I keep promising things, and then you could be asking me when is this going to happen. So the NCI is putting together a cancer data commons, and it's in, I will say, it's in the embryonic state. Okay, so it's going to be probably 3-4 years until it actually happens. But what they are trying to do is put together all these commons, the genomic commons, the proteomics commons, the imaging commons, the clinical data commons, all of that together in one site. So you will be able to do what the question just asked. Go from proteomics to RNA to the clinical data to the images if they exist. And so we understand that having silos is a problem, and we are trying to bring them, they'll still be silos, but they will have connections between them. And we're trying to bring them all to the same field. As you can imagine, this is quite complicated. And so we're doing good progress, but it's going to be a while. It's going to be, I'll say 3-4 years until we have that, but eventually we're going to get there, because we understand that people having to get data from different places to do an analysis can get complex for people that are not bioinformaticians.
So another way I would add to try to look at this mRNA and protein correlations without having to go to those sites to download would be, if you're interested in a particular tumor type and there is a flagship publications, those publications will most likely point to or have as supplementary tables a lot of summary tables of protein expression and mRNA expression, those that have been sort of rolled up to genes. So depending on the kind of analysis you want to do, that may be sufficient to just go to the summary tables that are supplementary to the publications.
Yeah, I think that's quite useful. And one related and last question for this session is, so when we get the protein sequence from CPTAC, how well does it match to the HUPO? Sorry, I don't know what the HUPO means. I think it's something related to
human proteome organization. So essentially, is the way the mass spec works is what you see coming out of the mass specs are peptides, okay? And then based on the peptide sequences, there are algorithms that will predict what those are more likely, what protein those are more likely to belong to. And this is how the measurement will be rolled up to proteins or genes. So most generally people are accustomed thinking of genes, but the reality is a gene can yield many proteins, right, that have different isoforms. So it depends on how the analysis is done. You may want to annotate all of your peptides that point to a protein to the gene where those are coming from. And that type of analysis, for instance, can be useful if you want to prioritize genes based on copy number, because you need to compare gene to gene, or you may use a different annotation where all those peptides are mapped to predicted protein isoforms instead of just the protein. So there are different ways in which annotations can be made. And then on top of that, the actual sequence of the peptides could actually be mutated compared to what's the reference, because that's an actual amino acid variant derived from a
tumor,
somatic mutation.
Yeah.
So I'm not exactly sure what that question refers to, but I guess the answer is it depends on the type of analysis and the annotation that you're using.
Yeah, absolutely. So the mutation could change the sequences as we see there. Okay. Thank you very much. We will come back to you if there are more questions.
My pleasure.
Yeah.
Yes.
Okay. So going back to Dr. Zinning again. So there are some questions related to the GDC portal. So the first question is about, is the pipeline available for offline analysis? Like, is it like a Docker or Singularity image one could download and use for their own data set?
So simple answer is not, and then the reason for that is, so on one hand, if you go to our GDC documentation, we tell users how we, the workflow, how we analyze those data. And in that workflow, each stop you have, in this stop we use the software from Broad, there is software from WashU, because we don't own the softwares. So you look at that workflow. If you're interested, I'm interested in Mutex, then you talk Broad. If you're interested Vascan, then you talk WashU, because we don't own their software. All the code developed by GDC is in GitHub. It's open, you can download. But that is more for like a data management system. But for actual analysis pipeline, we don't own it. And especially for like GATK, you have to have a license to use it. And the other, that's one thing. The other thing is a lot of our pipeline, like analysis pipeline, involve like a panel of normal. Because you have tumor, then you have normal. You want to filter all those, and those panel of normal is control access data. We cannot just redistribute it. But anybody want to repeat what we have, if you develop your own tool, compare with that, we tell you. From raw sequence data, we use BWMM, and you can download BWMM talking to the developers. And then we use 4 variant colors in each one from each organization. We tell you, and then follow up with how we filter those germlines, how we tell you that each step, how we do it. That is publicly available. Yeah. But actually, so we have like 5 boxes. A given box, because we don't own that box. You have to talk to the owner.
Okay, that's clear. Yeah. And the second question is about, can we search in GDC by a specific mutation and the genomic position? Is it possible? And then related to that is, if I have a set of mutation, can I get a frequency of that in the TCGA cohort?
So that you have a mapping issue. If you have a GDC mutation ID, you just go to search box. It will tell you all those things.
I see.
And then you can in GDC and not so in GDC the frequency of that mutation in the entire GDC, and also a fraction of that mutation in a given cohort. Say TCGA breast cancer, that mutation frequency in TCGA breast cancer, and that mutation in all breast cancer cases in GDC, and then you can compare with yours. So that you can get that, but you have to get the mutation ID from GDC first. It's chicken egg. So if you call your mutation, you have to find a way to define it. It's very difficult to reverse kind of map, you know. Or you have like a coordinates. We don't have search by coordinates.
I see. Okay. Thanks. We will come back to you if there are more questions. And for now, we move to Dr. Sia. Hi. We just have a few questions. I will start off with the first one about, the CPOA portal nicely represents different cohort. And it's quite curious to know how you do the initial QC and pre-processing for different data sets. Can we get access to that? Like as a user, can access to that? Like what are the steps that you apply for different cohorts?
So the CBI portal separates each cohort as you described. It's just called a study, and it usually is associated directly with a publication ID. So for the most part, most studies are just the processed data downloaded from a publication or from other sources like the GDC or PanCan Atlas projects, where the data is available, made available alongside the publication. And then that is just absorbed into the CBI portal by curators and programmers who convert it into the formats that the CBI portal can import. And that's about it, really. There's no additional processing or filtering that is usually done. Maybe if there are germline mutations and we're not allowed to show germline mutations in certain studies, that would be excluded. But for the most part, there is no filtering or anything done. The CBI portal by itself takes none of the responsibility of calling mutations or calling expression or calling amplifications and deletions. It leaves that to upstream pipelines. And then the results of that, it just specifies these are the formats we need the data in, and we can absorb that into the interface. And that's about it.
Okay. Yeah.
And for that reason, a big caveat is that each study is not necessarily consistently analyzed. And that's an important caveat. But there are nice shortcuts in the CBI portal that says choose all the TCGA PanCan Atlas studies. And in that case, you can have 32 studies that are guaranteed to be processed exactly the same way.
I see. Okay. I think it's a good point for everyone to be aware of. And the related question is like, as you said, each study will have a different data set they could have generated, for example, from genomics to mRNA, microRNA, and proteomics. Would all the data be integrated in the CBI portal, or it's mostly mutations and expression and copy number?
No, the TCGA data and some other studies where other data types were available does include so small substitutions in indels, copy number changes, fusions, RPPA-based protein expression, CpG island methylation assay data. So it's pretty flexible. For the most part, as long as if it's like expression data where you have a sample, gene level, a number per gene, right? As long as it's a number per gene per sample, you can name it whatever you want and put it into the CBI portal, and that explodes the number of the types of information you can incorporate into the CBI portal.
Okay, sounds good. So I have one last question, but this sounds very specific, but maybe you would understand what the user refers to. The question reads, when would MSK 2017 cohort release whole genome data for mutational signature analysis?
Whole genome data.
Yeah.
MSK does not have whole genome data. I mean,
see.
I guess we do. There is a pediatric cohort with whole genome data and matched RNA sequencing. Yeah, I'm pretty sure there is a study in the CBio portal that uses that publication. If I'm understanding right, that's the Eli Papamanuel lab paper. That wasn't in 2017. That was just this year. I don't like you. I don't really understand the question, so I can't.
Okay.
Yeah, if it is a publication the person is asking about, then it may or may not have been absorbed into the CBio portal, and I encourage them to contact.
I think
that would be great.
they need to contact MSK.
Yeah, exactly.
I think I know what they're talking about, but as Siri said, CBio portal does not expose germline. And as you know, when you do whole genome sequence, you have a lot of germline. And so I think that's why it's, again, the whole idea of CBio portal is to make all the open access data available. They don't have a control access tier. And so the whole genome sequence is very difficult to put in that box.
I see.
Yeah, that's true. You just made me remember the CBio portal does not have the ability to right now to show non-coding mutations in a meaningful way. Yeah, so that's also a limitation right now.
Yeah. Okay. Yeah, maybe we'll ask the user to contact you if they need more details.
Yeah, that's fine.
But I think it's all good. Yeah, thank you. So now moving to our next speaker, Dr. Gerald. Hey. Yeah, so we have a lot of questions related to Dragon here. So I think they got hooked onto that, and just a couple of related questions, I guess. So the first one is, how well the Dragon variant calling by using the GATK pipeline within the Dragon would match with the GATK open source? And how long this will be maintained as if there is any consistency?
Yeah, that's a great question. So we recently announced a collaboration with GATK together with Dragon. So the Dragon germline pipeline right now that's available is in collaboration with GATK. It's called the Dragon GATK pipeline. So we are continuing to work with the Broad Institute on that. And I don't think we have any time frame for the collaboration to end. And there are a lot of, we do have data showing benchmarking against each successive version that's out. And I encourage anyone who's interested to visit the Dragon website at the Illumina website. And we show version 3.5, 3.6, 3.7. 3.7 is the latest Dragon version, and it shows how it's constantly improving.
I see. Okay. Cool. And related to the other data types that you have presented, like the Dragon could handle, could you elaborate a bit on that? Like, what are the data types like, and how it fits with the other platforms like Nanopore or NanoString? If you get data from that, would Dragon could integrate those data as well?
Possibly. It depends on what level of analysis you're looking at. So Dragon is both hardware and software, right? And the software bits, there are different pipelines, but the very minimum that you can do is sort of just aligning, and it aligns very quickly. And I think I had one slide on how it uses graph to improve the alignment. So even if you're using sort of other platforms like Nanopore, I think that would still be applicable. And if you're looking at single cell data, we just launched a single cell pipeline. But of course, it might not be everything that you need. And the point of it is to speed up the secondary analysis part. And the tertiary analysis bits, you can sort of explore as you wish.
I see. Okay. And this is the last question, but again, this might be very complicated to answer. Will there be any open source version of this will be made available to users who cannot afford that big license?
That's an interesting question. So I think with the Dragon GTK pipeline, there might be a version that is being offered by the Pro that would be widely available. For the other pipelines, I mean, we do have sort of evaluation licenses that are available to users who wish to try these different pipelines out. So I encourage them to reach out to the Illumina Lab or Famous Life Sciences, which is the distributor in India, to discuss, and we're always happy to accommodate requests.
I see. Yeah, I think people are interested, and hopefully.
Great. Very glad to hear that.
Yeah. Okay. Thank you.
Thank you.
Yeah. Okay. So moving to the next speaker. Hi, Katie. We just have a couple of questions. Okay, so I think this is from your presentation when you have talked about the driver mutation. And the question is more broad, and they're asking how one can use the pan-cancer data set to identify driver mutation in a specific cancer. If they are specifically interested in cervical cancer.
In cervical cancer. So, one of the aspects of that paper from our Pan Cancer Project was to see, as now we expand our cohort and we're now looking at 10,000 tumors, what is our ability to find driver mutations? And so we were looking at it more in a pan-cancer context, and then looking to see how specific mutations fell out. So if you think about like your power to find recurrently mutated genes, it's dependent on the number of samples that you have. And if we're looking at something like cervical cancer, we have about, about 400, 500 there. And now we're looking at a little over 10,000. So we have the capability to find additional driver mutations that might be at, that wouldn't reach significance in a smaller subset. And so what I focused on in that when I was pulling in the driver mutation data is to show that really there's not that many driver mutations, even when we're looking across 10,000 tumors. We had just shy of 300. And what was really interesting is not that many of them were actually found across multiple tumor types. So only, was it like 50 or 60 got picked up as being found in multiple different tumor types. And the vast majority of them were cancer type specific. And I think that's, particularly interesting if we think about like the ATAC-seq paper that I mentioned, there's a lot of tissue specificity, and there's potentially, how either the chromatin or the methylation is regulating where you can potentially have mutations. And so, there's definitely ability to take, just because we broke down the driver mutations of whether it was found in multiple cancers or only could be picked up if we looked at 10,000 cancers, I don't think necessarily says that those drivers aren't important in a specific tumor type. I think it just was a way for us to have a larger cohort to have more confidence in finding rare potential driver mutations. But you can totally take that list and extract out the mutations that you think are potential drivers within, just a subset of that cohort. But I think, the cool thing that I thought from that is that there really aren't that many driver mutations, and they aren't that frequent. And so when mutations are important, it's the combination of all the different types of mutations and all the other different types of alterations that are leading to, the diseases and the heterogeneity that we're seeing both within and across different tumor types.
Yeah, that sounds good. And is there any way where we could access all this kind of more analyzed data, like finding the drivers or multiple alterations? Can we get it in the GDC kind of thing? Like these are all the core things to look after. Yeah.
So the driver mutation data came from work that Kyle helped lead from our MC3 data, and actually SARIAC too, multi-center mutation calling effort. And they have generated a public version of that is just the coding mutation. So it doesn't have the non-coding, and because we moved out anything that could have any potential for germline. But all of that coding, so mutations that are within the exons is a publicly available file. And I put a link, I think, to our GDC publication page where you can find that math and can be downloaded for analysis. I won't lie, it is huge because it is all 11,000 tumors. Some of these are hypermutators that have, you know, hundreds and hundreds of mutations, but we have that data available. It's public. And I think, you know, what I really like to stress is there's the raw data and kind of some process data at GDC, but these publication pages that we've hosted on GDC have a huge wealth of data with stuff that you don't have to reinvent the wheel. You know, we've got those mutations, we've got purity and ploidy estimates from copy number, we've got the, you know, GISTIC outputs and segment files if you're interested in copy number, the gene expression matrix and microRNA expression matrix that have done some of these batch normalizations for you, because TCGA started in 2006 and we finished processing data in 2015. And as you can imagine, considering we didn't even start with next generation sequencing when we started in the pilot phases, all the transitions and sequencing that happened while we were active, we've gone through some of that hassle. And I want to say we spent 2 years just like checking these samples and figuring out what was right and trying to do some batch corrections. And so we have that data available for people who don't want to go through that hassle on their own. For people who want to come up with their own methods and do it their own way and maybe better, we got that data too, and you can totally use that as well. But I think, you know, the resources that we've provided of this more processed, analyzed data on our publication pages is a fantastic resource for people to use, and it includes those driver mutations.
Okay.
Yeah, and I will emphasize that, you know, we have all this process data that is available freely. Anybody can go get the files. And it was a lot of work. MC3 was supposed to take 6 months. It took 2 years plus because it's a very complicated process. So why are you going to do it again? You know, the data is there. Use the data that we already took the painful step to clean up. And you know, it's available. And again, that goes to what many of the questions are, oh, you have this control access data. It's like all of that, all of the supplemental files that are in the publication pages, all of that is the raw data that we use to display the GDC data. And so if you want to go to the raw data and apply your methods to it, that's the raw data you want. You don't want the sequence. You know, you have to be really a sequencing geek to want the sequence. But as Katie was saying, it's like we have all these pre-processed, cleaned, batch effect corrected data. That is what you should be using, not the original stuff.
Yeah, I think that's why the process data is available, and they should go for that for a safety.
So this is any one thing. So when you go to GDC, either GDC portal or if you go to the GDC website, both of them, don't forget upper right corner. On the upper right corner, it says GDC apps. Click that. There's publication page link. Then you have all the TCGA marker papers and their supplemented. Of course, you have all the PanCan analysis papers and the associated data. So it's there. Upper right corner, GDC apps publication page. Do you have anything about publication of TCGA and PanCan analysis?
Yeah, I think that's very useful. Yeah.
Yeah, and I will say one thing. So, you know, TCGA happened in the early 2000s, and, you know, we finished our publication like 2018. Everything that we did was in HD19. And the new harmonized data and processing pipelines that are in GDC are all in HD38, and there's updated pipelines for many of these. And so that is one thing to keep in mind, because I had questions from users who are like, the current HD38 data is not matching. Well, there's some differences. And we do have a publication that we put out that compared the harmonized data with the prior legacy TCGA data that is available, and kind of walks through some of the differences. And the reality is they're way more alike than they are different, but there are some, you know, key things to keep in mind. And we've outlined many of those in this publication that really kind of looks at the differences between changing the genomes in some of the cases where we had to change the alignments, the quantification metrics, or the, RNA had probably the most changes because we had everything changed. But I think that is a good resource and something just to keep in mind that, taking the old legacy data and the new harmonized data, that there are some potential differences since we switched genomes. But just, keeping that aware, but we try to make as much data public and as much data, usable data, that people can just grab and use for their own analyses.
Yeah, that sounds good, and there is a relevant question related to that, and it's about regarding the methylation. Sorry, this question is supposed to be for Dr. Berman, but I will just post it here. So the user is interested to know how you have managed to convert the methylation probe that mapped to HG19 to HG38.
Oh, I wish we had Ben or Hui or Peter on. I believe they did. Did they remap them, or they did liftover of the region?
So they did a liftover of the data. But again, things that to look in the future. We have now the Sesame pipeline in the GDC. And so what we did a liftover from the old calculations to HG38, but it is in our list of things to do to reprocess all the files using Sesame. So eventually, everything will not be a liftover, but actually calculated on 38 on the original files. Again, eventually, we have a large list of to-do things. So, and we have so much bandwidth, but this is something that we have. And that also goes for, but Katie said there were so many things that change in expression, which is true. We have the new pipelines. And so that also goes for expression. Eventually, we will run all the old files that we have with the new pipeline, and everything will look the same. But it's a high lift. It is one of the requirements to the GDC to do. So again, at some point it will happen.
Okay, sounds good. Yeah, and one last question to Katie. So you have presented this cluster for the breast cancer sample based on the expression. And the related question was, if you want to go for the survival analysis, how much power do we have if one have to do the survival analysis for the different clusters? Does it make sense?
What type of analysis?
Like, I think there was a slide where you have clustered the samples based on the expression in breast cancer and related to the cell type as well. And if one has to do some survival analysis, specifically for each of the cluster, how much power do we have to check if the expression of one gene in one cluster has a better prognosis or not?
So single gene prognostic for looking at outcomes or something like that?
Yeah.
One of the things that we found is that single genes typically don't tend to be as prognostic as gene signatures. And so finding, groups of genes that are that they tend to behave better than a single gene on their own. And in terms of our breast cancer subtypes, we've found in multiple cohorts and over multiple years that the classifications, and whether they use the ones that I was trained with or some of the other public ones, is that there are some pretty strong prognostic capabilities of these subtypes. So, basal or triple negative. And from what we saw looking across multiple different data types, really does look like it's probably a different disease, a different etiology. It's in younger women. It's more likely to be in Black women at a younger age. There's different risk factors than we see for estrogen receptor positive breast cancer. So where breastfeeding is protective for most breast cancers, it's potentially a risk factor in basal-like breast cancer. So, and we see that whether we use copy number, mutations, gene expression, DNA methylation, microRNAs, they look incredibly different from the others. And when we use expression signatures of those groups, we find that they are prognostic and they indicate a worse outcome tumor. In some of our other subtypes, like the HER2 was a pretty poor outcome, but tumor in early cohorts pre HER2 targeting drugs. But now we see that when we can identify that group and treat them with the appropriate drugs, their outcomes are actually better than what we observed 15, 20 years ago. So I think in terms of individual gene prognostics, there's definitely genes that are going to be prognostic, but I find that signatures tend to be better than a single gene. And I find that when, one of the things I think from PanCan that was really interesting is when you find subsets of samples or tumors that look distinct by multiple different data platforms, it really does suggest that they probably are a unique entity that really is not going to behave like other samples from that same organ, and really shows that the heterogeneity sometimes within a tumor type is just as large as the heterogeneity across tumor types. And I think that was something that was really interesting as we looked across now 10,000 tumors and not just restricted within a particular single organ site. That was quite informative, and the fact that there was so much tissue driven groups. And we often get the question like, oh, well, it's because you're using bulk tumor, we're probably picking up the microenvironment. But the thing is that we get that for the DNA methylation and we get it for the copy number. And those, by having non-tumor components, actually just kind of reduces a bit of that signal when you're looking at things like methylation and mutations. And so I think there is actually very strong tissue components in some of these cancer types. And you can see it in the types of mutations, like why do you only see APC mutations in colorectal cancers? There's clearly tissue specific reasons. Certain alterations are occurring. And I think, understanding kind of those aspects is important. I totally deviated from a single gene, but hopefully, I think there's so much cool stuff being able to understand a particular tumor type by now looking across other tumor types to really get understanding is, how much variability do you observe within a particular cancer?
Okay.
Yeah,
Cool.
and one comment on that is, they ask her about the driver genes and the driver genes and the driver gene. I hate that word because that gives you the impression that the tumor is the consequence of one or two genes, and then the rest are there for the ride. It's not true, because the same patient can have the same two mutations and react totally different to therapy. And that has to do with the whole component of the genomic background that they carry with them. And so, we're having all these clinical trials in which there's this one that is like the archetype of my point, which is the one with for melanoma, that for BRAF, they targeted, and every single patient in that trial had the same mutation. 85% of the patients in the trial responded. Okay, so why did the other 15 not respond if they had the right mutation? Well, they didn't investigate that. Okay, and then out of those 85% that responded, 20% responded long-term. Okay, everybody has the same mutation. Why is 65% not responding long-term? And so you see, we are missing the forest because of that single tree. And so I'm totally with Katie. We should evolve and start fixating on single gene issues, because that does not help. Cancer is very, very complicated. It doesn't happen because of a single gene. So I will plead with the audience to move away from the idea. It's an appealing idea. If you have a single gene and you have a therapy for that single gene, ta-da, you have the solution. But it's clearly not the case. And so we need to move away from that.
Yeah.
This is a perfect opportunity for me to plug in the value of looking beyond just the genomic sequence to try to find, signal transduction pathways, some other features that are associated with response or resistance to treatment, which is also part of what CPTAC is embarked on.
Yeah,
and,
absolutely.
I was about to say there's a ton of value now that we have ability to look at whole proteomics, which we didn't have 5 years ago, or to the same extent that we do now. And so I think, CPTAC is really going to add extensive value to what we've done in TCGA and what was done prior in the Human Genome Project as we start layering in all these different data types.
Yeah, makes sense.
Thank you, Katie.
Yeah. just motivated by this, I just want to ask one question from my side. So in the long run, if we have to take this to the clinic, and let's say that we have a clinic where we cannot afford many of these different types, how one could make it to one, like a minimal set where I could pay less and then...
with that question.
Can I take that?
So we are actually running a project in which we are taking all the classifiers that we have done for TCGA, because there's tens of thousands of them, and we understand that the clinics can not even in medium or low resource country, in high resource country, clinics cannot test that. There's no way we can use signatures like that. And so we are shrinking the signatures. The whole idea is, and the challenge that I gave the group is, I want a set of genes, methylation, expression, mutation, whatever it is, that will define these subgroups for an assay that carries no more than 1,000 markers for all 33 tumor types. And I'm glad to tell you that we're very close to a solution for that. And the whole idea is to then transport that into a system that is portable, easy to assay anywhere using nanotechnology. And so when I describe these, people sometimes say, oh, you want a tricorder for cancer, you know, like Star Trek, you know, that scanning thing. I go, that's exactly what I want. I want something that works on 2 AA batteries, and, you know, and it uses just spit or your blood or whatever it is. And it's easy, and we can do it in the middle of New York or in the middle of the Sahara, you know? And so we are very much aware that the classifiers that we put together in TCGA are not ready for the clinic, but we are trying to get there. And we have that program has been going for 3.5 years. It's a very complicated thing to do. It's called the Tumor Molecular Pathology program, and but we're getting close. At the beginning, when I posed that challenge to the network, people were like, "Oh my, no, it's impossible. We cannot do this." And then, you know, everything is possible. It took a while to figure out how to do it, but stay tuned. Basically, we're gonna come out with a publication saying, if you want to classify the tumors, these are the markers that you should use. And the challenge is, all of them can be measured with a BIV or with a nanotechnology system. And so it's not something that you're going to have, you know, full sequencing power. No. This could be done with nanostring. And so we're working on that.
That's very exciting. Looking forward to that.
If I can add on the proteomic side, then the beauty of the mass spectrometer is that really you could, it's very scalable. So right now there are methods that have been optimized to be used with a single curl of an FFPE section. And it all depends on the number of proteins that you want to be able to assess, but you can always assess what's called comprehensive, which is whatever you can see with that amount of tissue, right? Mm-hmm. And so part of the reason, or a big reason, that the CPTAC is putting this data out in the public domain is so that people can start understanding the biology and making predictions. And so you can either go into the clinic with a limited resource sort of tissue to look at comprehensive within whatever amount that you have, or apply methods to determine a minimal set of analytes, or a minimal set of proteins that you want to analyze, and come up with a targeted mass spec assay that will interrogate a panel of 10 or 50 proteins. And for instance, one of our groups is generating a panel that is called an immune oncology panel, or there's another one that is a DNA damage response panel. So you can attack those and look interrogate those pathways within a cancer type or come up with your own particular panel. This is also part of what's being looked at to assess response once you know what it is that you want to be looking at. And so if people are interested in fit for purpose assays, we have those assays that have already been pre-developed in the website. And also the things to think about there is to start moving into an environment where the particular assay is approved by a regulatory entity. So we try to move sort of mass spec into that so-called CLIA environment as well. And sort of in the talk, I think it's tomorrow, by Henry Rodriguez, he will talk about that particular aspect as well.
Sounds good. Very valuable and looking forward to that. So in the context of the biomarkers, there is a question to Kyle. So you have presented the biomarker prediction. And one of the question is like, is any of the marker being used for clinical trials in any of these patients, and how that information has been or has been used to refine the biomarker prediction?
So it's exactly where JC had just described it, in that it is an in-process analysis. And basically the high level of what I was talking about was actually the TMP project. That a majority of what we consider to be, important for the, usage, like biomarker prediction, is this subtype analysis, and I've been able to identify all of the different subtypes. And so I concentrated in my talk, I concentrated on breast cancer, but we're basically doing the exact same analysis across all of the tumor sets where there were enough samples to actually do machine learning. Now, that is one of the caveats on that statement, is there's some cancers where, there's just not a large enough population to train a predictor effectively. You just don't have enough samples, and that's one of the rate limiting factors. And to Jason's point again, the thing that we're really kind of interested in is what data modalities provide the most information with the least number of biomarkers. And so we've spent a lot of time on expression because it, I think I talked about like mutations, they occur, but not regularly and all over the place. And you don't even know if the mutation you have is even having an effect. It could just be like a silent mutation that doesn't actually do anything. It just kind of mirrors something you thought was important. But like looking at the expression and pulling markers of the downstream effects is more informative. And interestingly, and truly the question of refinement, we're not even sure if the biomarkers we're pulling have anything to do with biology itself. They're just indicative of the subtype. They're not actually part of what we would consider to be basic biology. They don't tell us anything new about the tumor other than how it's behaving. But you can't look at the gene list that we're coming up with and saying, oh, that gene does this. No, it's just, a lot of these are just correlated with the event and therefore happen to be very effective for prediction. And the machine learning algorithms pick up on them. But that doesn't mean that they're actually, telling you anything about treatment or anything about biology other than helping you identify the subtype. So they're what we find is only specific to the problem at hand, the task that we're doing, and only does that. And it doesn't, you can't like extrapolate more biology from that.
I see. Good. Just don't take me wrong. It's just a personal question. Like you were saying that the mutation is not doing anything, but
okay, the mutations.
But people going for the immunotherapy or designing vaccines by the type of mutations they are presented in the neoantigens.
Well, when I say that, I don't think he said that the mutations are not relevant. But the thing is, if you focus on the mutations, you're kind of killing your power because it does not have that much of an effect. And in fact, what he was saying about the biomarkers not being relevant for biology is very clear, because when we started this, we had all these markers. And then when they started doing the analysis, they go, oh, all of these markers actually are saying the same thing. So you could pick any of those to be part of your signature. And that means that they're not what is causing the problem. They're just it's like a flag that it's saying it is a problem, but they're not the causal reason. And so that was a very interesting thing that came out of this exercise.
I liken it to being able to spot smoke. We're looking for smoke, but not the actual fire.
I see. Okay.
And again, to the mutation point, I'm not saying mutations are incredibly powerful. The problem is that they occur all over the place. And if you only concentrate on one band, and this is the problem with biomarkers, is that you have to concentrate on a single thing. And if you can only concentrate on one thing, and it occurs, you know, like mutations can occur anywhere in the gene, and they could even occur in the translational area above in front of the gene. You know, it could have like a promoter region in front of the gene, and if that is not part of your assay, you're going to miss it. And so it's very easy to miss things when you have narrow bands. And that's what biomarkers are, is just looking at very specific things. And the problem is the mutations are not one thing. They're actually a multitude of possible events. And we call them one thing. We say, oh, it's that one gene. But actually, you know, I think I included a link to like the BRCA mutation database. It is a database of over 40,000 mutations for one gene. And interpreting that is kind of, you know, very difficult and not what I would call a biomarker per se. It is an analytical problem.
I see. Okay, so it's more of a challenge. Okay. And we have one more question, but I think this is very specific to one of the slides that you have presented, where you have defined features for the classification. And I think you probably have mentioned n feature equal to 50 or something. So how do you come up with these numbers for feature selection?
The budget that JC mentioned. No, that's my joke. Well, it just works out that way. Is if you want 1,000 and you have 20 cancers.
Jesse get it well.
Kind of what? Okay, so I said we have 33 tumor types. Let's do 1,000 markers, and I remember Paul Spellman asking me why 1,000. I said I don't know. It's a nice round number. You know, it's. You know,
we did find that
you have to constrain yourself to what is possible to analyze, and you know, in a logical budgetary way, and so that was it. But yeah,
we did find for some cancers you can actually get away with much less than that. I think we did have somebody who turned in like signatures that had like 100, and they did slightly better, but not much better. You know. And this is all in the standard deviation noise level, like, okay, you did 1% better, but the standard deviation is 3%. So do I really, you know, does it really make a difference? So it's a diminishing returns thing. You know, you can keep on adding features and features and features and always get slightly, maybe incrementally better, but it's a diminishing return. We found that, you know, I think I'd presented that RFE analysis where it's just like, okay, we keep on dropping features iteratively until your signal goes to nothing. And I think a lot of them, they kept in a nice plateau down to like 50 or even 30 for most cancer types. So you can get majority of the signal, at least, you know, from what we've seen, for most cancer types down to that much. And so we had effective predictors at, you know, with 50 or less features for almost all of the cancers. And I think the cancers where we didn't, it didn't really even matter, because even if we added a whole bunch of features, they were still hard to predict anyway.
It all depends. Go, Caesar.
Were those most likely to be methylation features, or do you know?
No, everything came up as expression. It was
expression.
Yeah, because
no mutations.
It's a bias of the machine learning algorithms. I think the way that they treat numbers, expression numbers are just, they're getting more signal. Methylation, the problem is that you start out with like 500,000 probes, and then you have to filter down. And feature selection is the biggest problem here, is picking out which features you're going to concentrate on and pass over to the machine learning. And so if you don't do that correctly, or like some people do this feature selection and machine learning at the same time almost, but it's a very difficult process to kind of filter out what you don't need to know about. And some, like I said, 500,000 methylation probes to start out with, and they operate in islands. And I think so it was, yes.
Sorry, well, that's some
Obviously, to
weightage.
translate to the clinic, having a expression-based system helps because it's a lot easier to do. And what Kyle was saying is that it depends a lot on the tumor type. So there's for example thyroid tumors. They're very simple. They're genomically quiescent. They just don't have a lot of signal. And in fact, some of the models have basically two features that define, the subtypes for the thyroid models, and that is logical because it's a simple one. But then if you go to lung, well, that's another ball of yarn, and so it all depends on how complex the tumor itself is.
Yeah, my comments in terms of like expression, that was for a majority of cancer types. That was not all cancer types. I think melanomas was very much driven. The subtyping is based off of mutations, and the best predictors were based off of mutations. That was just, but for that specific cancer type, and I forget which ones did methylation, but there was, I think, one or two where methylation was the principal component
for gastric. You need methylation because otherwise that fourth subgroup doesn't show up.
Yeah, exactly. So again, I'm saying a lot, but not all.
Yeah.
And to some extent, some mutations and some methylation change will result in expression pattern change. But I do have a question for Kyle. Do you also look at you look at just gene level expression, or you also look at alternative splicing?
For this particular, no, we did not look at alternate splicing, and I understand that is a weakness. But again, I think sometimes the alternate splicing is going to the specific biology. If we, again, that's in a specific cancer event. But if there is a downstream effect that is resultant of that specific alternate splicing, we can pick up on that signal. So,
Guraj, you had a comment?
Yeah,
what was that?
Anguraj, you had a comment,
Anguraj,
right?
Comment.
Go ahead.
Yeah. So I have a question, a couple of questions. This is very interesting discussion. So is it like a pooled pan-cancer samples that you were analyzing, number one? And number two, is there a weightage added to the machine learning approach that influences the gene expression?
So what was the second half of that question?
So the second question is there any weightage or anything added to the machine learning that influences the gene expression to be the most important biomarker that comes out of it?
So yeah, it was the pan-cancer set. It included a subselection of mutations, microRNA, mRNA, copy number events, and methylations, but the methylations have been filtered by variants and some other stuff just to only pick subset out, so we didn't deal with a full 500,000 methylation sites. In terms of weighting, I don't believe there was anything weighted to expression other than a possibly numerically, it looks more like the kind of data that a lot of the machine learning algorithms tend to do well with. Mutation information comes across as a binary matrix. And so it's just a bunch of ones and zeros, mostly zeros with just an occasional one. And machine learning algorithms don't do well with that kind of stuff. So I think there is implicit bias, not on the weighting of the different data types per se, but more in terms of how they get interpreted to the machine learning algorithm. And I think there's some bias there.
The reason why I'm asking is that we also see that RNA-seq most of the time gene expression dominates. It's completely different data set other than TCGA, but it also varies depending on what kind of data set, and secondly, the number of features. Most of the time with the RNA expression, you will have more number of features. That probably, I don't know, depending on the machine learning approach, it probably kind of goes for some weightage, increased weightage that probably starts preferring. So that's why I'm just trying to understand that.
No,
anyway.
There's even fun effects like, making sure that you normalize numbers correctly. Because like if you express your RNA is in like, RPKMs or something like that, it goes from your dynamic range is huge. And then in another data set like copy number, this dynamic range is small. And if you don't weight that correctly, then you jump it all into a machine learning algorithm. It'll always go for the features that have the largest dynamic range, because they're the just like multiply in the math and make the biggest changes. So stuff like that, it's not even part of biology. It's part of just your dumb translation or representation of biology into math for the machine learning. And if you don't do that correctly, you can end up with weird effects like that. And I think just, the way the numbers are shaped for RNA or expression analysis, they work better for machine learning just because of that, the dynamic range a lot of times.
Sorry, last one question related to that. So do you scale all these multi-omics features together? I understand that it's not easy, especially when you have a binary variable. So did you try them as well, some kind of scaling of all the multi-omics
So
data?
what we did actually was, again, this is going to the TMP working group, was treat it as a challenge. So there was actually 5 different teams that were actually tasked with the problem, and we just let them all compete. So whether or not they did or did not include that was part of their pipeline. And if that improved their ability to create realistic models, then that got included in the work. So yeah, we just challenged it and just let people, the different subgroups,
I understand.
Figure
I
out the
think
best
it's
ways.
challenging. Yeah, thank you.
Yeah, I think this is a very interesting discussion, and I think this is quite good learning for others too, who wants to step into the machine learning, since it's exciting. Okay, thanks, Kyle. I'm just moving to the next speaker. Dr. Min, we have some questions related to the protocols and the details. So
I can help there, yeah.
Yeah, so the first question is that, there is any cheat sheet available where one can find how much tumor is required to reach this many number of cells for different tissue type?
Yeah, I think that's a very specific question. I think before I start answering, I'd like to say thank you for having us here, and also to say really excited that I heard early on in the session that TCGA is moving on to the single cell realm. So looking forward to those data sets. To really answer the question, I think it really depends, as the panel has spoken a little bit, on the tumor type itself. So it's very difficult to give a ballpark, but feel free to reach out to us. We also have some estimates on our website that we can point you to. So if you could link me up or us up with the person who asked the question, definitely happy to move forward with that.
Sure.
In the future,
yeah, sounds good. And related to the protocol, it's just wondering why Chromium is used in the protocol, and what is their significance and what it does actually?
I think it's a rather broad question, and I think if you look at it, Chromium, I would say, is our platform, right? So it's actually the instrument that does the partitioning of the nanoliter gems, right? So you require that workflow to partition the gems, right? And that will be able to get you to the libraries. Now, so you either have the Chromium, you either have a Chromium controller, right? The instrument that packages all the gems, right? For you to do your first part of a library preparation. And thereafter you do your manual NGS library preparation. Or you have the Chromium Connect, which then is an automated system. It consists of a Hamilton robot, which then does the library preparation for you. So in a sense, it's kind of a name for the method, so to speak.
Okay, so there is no Chromium being added?
No, that's a good question. No, there is no Chromium added to it.
Okay.
I think
that's I don't know. I'm reading the question literally, so
okay.
I just wanted to get it clarified. Okay.
That's an interesting question. Yeah, thanks for that.
Yeah. And the other question is about, is 10x planning to introduce single cell ChIP-seq protocol?
That's a good question. I believe we are not there yet. We have been having a lot of requests, and I think what we've just released earlier this summer on the northern hemisphere. But what we just released would be an ATAC-seq, single cell ATAC-seq plus gene expression from the same single nucleus, right? So as always, when we first launched single cell ATAC-seq, the first question that came in were, are we going to be able to do gene expression studies on that same nuclei, right? And from what we hear, one of the first few questions after we launched ATAC-seq plus gene expression was, are you going to have protein expression on the same cells? So it's always an iterative, I guess, a process. We do hear requests on ChIP-seq, but I think part of that equation is it's very, very specific to all the different antibodies, which is probably why I can't say on behalf of 10x whether how far we are in that protocol, but we did look at it. I'm not sure if you can add a bit on any timelines, or I can review any timelines, so to speak.
Yeah. But having said that,
I must say, if I may comment,
yeah, please.
For people that are thinking on ChIP-seq, be happy with the ATAC-seq. You get pretty much the same information with a lot less trouble and problems with, you know, ChIP-seq is very laboratory dependent. It's very, I would say, technician dependent. You know, it's like the person pipetting makes a difference. ATAC-seq is a lot more stable, and you get a lot more consistent results. So I know that people are accustomed to ChIP-seq, but you know, ATAC-seq gives you, for the most part, the same type of information with a lot more reliability. So I will be very happy that the next has an ATAC-seq protocol and use that.
Yeah.
Thank you so much for your support. And I think from my personal experience, I used to do a lot of RIP-seq and microRNA pull downs and so on. And I agree with you. It's very, you know, technique dependent, and of course for ChIP-seq, it's very antibody dependent as well.
Yeah.
Yeah.
And we know how reliable the batches from antibody from for the same antibody are from batch to batch, and so you know,
right.
It's quite a problem, really.
Yeah. Sounds good. And also the combined ATAC-seq and RNA-seq is very exciting too. We're looking forward to that, and hope the budget won't be too high, given that both have been combined. So
yeah, what I can tell you that is well, it's 1 + 1 doesn't equals to 2. So it is cheaper than a double. Yeah.
Okay. Looking forward to that.
And if more people use it, it will become cheaper and cheaper as, you know, sequencing. When we started TCGA, we couldn't afford actual sequencing. We had to do targeted sequencing because it was incredibly expensive. And as we commented, we are doing, you know, whole genome sequence on every single sample. So, you know, eventually, you know, with use and technology becomes cheaper.
Again, appreciate all your
support.
support.
We all want everything now and for free.
No.
I understand. I've been there before.
That wasn't my question. It was more affordable. Yeah. Yeah. Okay. Thank you. One last question. Again, I think this is very specific, but you see if you could relate to that. And the question is, how does Visium differ from SciSafe developed by Brigham Women's Boston?
Unfortunately, I'm not very familiar with that protocol. If you could describe that protocol a little bit to me, I can describe a little bit on the Visium side of things, right? I think and compared to commercial alternatives out there, right? The main difference is that we have, if you're looking at a single tissue section, we are looking at the spot size, which is very small, about 55 microns. So that's between 1 to 10 cells. And I think that's very that's quite a small resolution. The second thing is that we are able to look at it at the whole transcriptome level with the poly A pull downs or with the poly A identification. And the third is we are able to look at the entire sample at the same time without having to isolate certain elements and say pulling out the RNA that you're interested in and then sequencing. So we do the whole slide at the same time. So I will need to review the exact what do you call it technology that you mentioned.
Yeah.
But that's basically our three. I think the last thing I want to mention as well for Visium is that you do not need an instrument from the 10x side, right? Basically all you need would be that slide that I showed in the presentation for those of you who attended, and that's basically the secret sauce, right? The Visium slide, and then that's all you need from our side.
Okay, sounds good. Thank you. And if the participant needs more details, we will ask them to contact you.
Yes, please feel free to try. Thank you.
Thank you.
Well, since I'm here and he's here, I'm going to ask a question.
Sure.
Okay.
So I know that you do poly A tail drop for this, which is fine. We use poly A tail drop forever. Any plans on going to something akin ribodepletion and looking at immature versions and long non-coding that don't have poly A tails, anything like that? And if possible, you could have it next week and for like $2.
So what I can share with you is that we if you look at the single cell side, it is very poly A dependent. I don't think we are looking at a non-poly A version on that front. But what is really exciting is we have announced Visium for FFPE samples that's going to come out early next year. And because FFPE sample is so fragmented, for that protocol, it's not going to be poly A dependent, but it's going to be a probe-based kind of approach, right? And by that, I think you can extrapolate and imagine that it could
you
be
could do
non-poly
probes
A
for
band.
anything that you want. Yeah,
yes.
absolutely.
Yes.
Absolutely. Okay.
Yes.
Thank
Hope
you.
that helps. Yeah.
Okay. Thank you. Yeah, and now we're going to Anguraj. Hi. Okay, cool. We are quite excited about this global oncology program that you described. Congratulations on that. And could you explain a bit of how we as a ICGA could get connect to that? And what are the the lessons that we could take forward?
Sure. So the interest with the global oncology is to see how the Institute of Cancer Research, as an individual institute, coordinate multiple activities across different low and middle income country-based research. One of the motivation for this is because there are, ICR as a whole has about 80 to 85 teams, but within that, around 10 teams or more are actively involved in these kind of activities. So that's the major reason why we initiated the Center for Global Oncology at the ICR, number one. Number two, we also coordinating this as a part of the London activities that's happening around. For example, with Imperial College and ICR, started a new center for epidemiology and prevention, just launched a couple of weeks back, and be connecting to it so that, we are just reaching our neighbor. That's number one, two. And number three is the UK-based Global Cancer Network, again, was launched just 2 weeks back again. That is the first global oncology program that has been started, involving very interesting stakeholders as a part of this. So, the idea here is how do we extend this as a part of the whole UK. So having said that, so this just kind of connects one to the other and puts them all trying to put them all together. So how ICGA, the Indian Cancer Genome Atlas program, could connect, that's already happening. Santosh should be probably listening, I guess. So the whole part is ICR already part of the Indian Cancer Genome Atlas project. We got the approval for that, and in multiple ways we are just trying to do this. But this whole point is not just the ICR alone, but what are the resources from the UK that can probably be provided to the ICGA to promote this. I can see that there are many people, especially TCGA itself, jumping in, helping, and that's the motivation for the ICGA. And this is an idea for how UK can participate, and eventually how Europe could also provide certain aspects of it for the ICGA to move forward. That's the whole idea.
Okay, sounds good.
Hope that answers your question.
Yeah, I have a related question. I will come back to that later, which involves everyone in that. So related to your presentation, you talked about the machine learning thing. So how we can use that to impute some of the data set that we could not produce? Is that possible? Like, for example, I could afford only 2 out of the 5 omics that we plan to do, but eventually we didn't reach there. We got only 3. Is it possible to impute some of the remaining based on the data that is already available?
So the answer to the question is yes and no, depending on what kind of quality data that you need. Definitely, there are others who could also pitch into it. So from our perspective, the machine learning approaches that we develop, we try not to impute from one omics data to the other so far, but there are other methods, like, for example, MOFA, which is getting popular as well. they try to do that. So what the consequence of that, we don't know yet. We are just experimenting currently. The answer to your question is yes, it is possible. If there are some data missing as a whole for one subset of samples for one omics data, people are imputing it. But I would be glad to hear from the TCGA colleagues that, what that means and how that would affect, because still the consequence of that, at least I'm not aware of that.
Okay, sounds good. I think there is one more question that I just putting up. So there are some questions from the users while we were talking. I think Ankita is compiling those. Ankita, do you have any questions? And while you are connecting, I have a quick question for Syriac. In CBio portal, is there any option to do signature analysis, mutational signature analysis, or gene expression signature analysis?
I have a fuzzy recollection that they were working on something like that, but it is input you have to provide so it can then visualize it. I don't think you can actually do the signature analysis on the CBio portal given a list of mutations. They were planning to, so at the best you can insert that information into the clinical data fields per sample, or the sample data. So if you've pre-calculated what the signature distribution is, you can just add it to the predominant signature. You can add it to the metadata per sample. That's one way to do it. I do think that they were planning to look at the mutation data that is uploaded to the CBio, imported into the CBio portal, and then based on the few mutations there, model what the predominant signatures could be. But I don't know if that's implemented yet. I could find out. If the person reaches out to me, I can find out and answer them.
Sure, we'll intimate them. Ankita, are you here?
Yeah. I don't see any questions.
Okay.
Live session.
Okay.
So
Okay. If there is no questions, I will just wrap up with one last question. And before that, thank you all for joining and for your time and patiently listening to me reading out all these questions and the wonderful discussion. It was very useful. As organizers together, we would like to hear your feedbacks or any input for the ICGCA proposal that has been put out, which is the Indian Genome Consortium. And we would be happy to hear any feedbacks or the lessons that we have to keep our eye open kind of thing. If it's too much to talk here, we will be happy to hear it later too.
Well, I have been conscripted to do that. Respond that question in 2 days. But I think the first thing I would like to say is I cannot believe how far you guys have come in a year. From thinking about doing it to actually having government commitment, having large participants coming to the table, large participants that sometimes don't talk to each other, and agreeing on doing this. It's I don't know, when we were discussing this a year ago, I thought, yes, you have to do it. Yes, it's a great idea. God only knows how long it's going to take. And I have to commend all of you because you have done in a year what TCGA took 3 years to do. Obviously, you have some experience for what the projects were before, but still, I was not expecting how fast this pilot is going. So I'm very happy, and I'm very happy to hear also that it's beyond not the Indian subcontinent, but beyond the India national boundaries, basically, that the Bangladeshi government has said, oh, this is something worth doing. and they are embarking on it. This posed very well. As I was talking to some of the leaders yesterday, I think the moment you have one project published is the moment that you're going to start hearing from other countries in Southeast Asia, because I have been talking to people in Burma, I have been talking to people in Thailand, and everybody says, oh, we want to do this, but how difficult it is to do here. And so I think the moment you guys have the first publication saying, hey, here, we did one, and we're going for more, I think you're going to have a lot of more people coming to you and saying, how about if we do this together? And I think that's the way it should be, because as I said last year, TCGA was done not for the American public, it was done for humanity. But we need TCGAs everywhere, because the American makeup is not the makeup of the world, and so we need people doing this type of analysis in different ethnic backgrounds, and I think India has a great opportunity there to look at something that we couldn't, which is what is nature versus nurture. Okay, you have a large Indian diaspora all over the world, and so you have the opportunity to see what happens to those people of Indian heritage that have, say, moved to the UK, moved to the US. They have a different diet. Yes, they maintain, I love parathas. Everybody eats parathas, fine, but they're not the same, okay? And the spices are not the same, and the environmental exposures are not the same. And so I think you have an opportunity here to have a really very interesting project that shows not only what ethnic backgrounds contribute to cancer, but also what your environment and what your present living conditions contribute to it. So, and I don't think, other than maybe doing this in the same scale in China, I don't think there's any other group that has this opportunity, that people that have left the country and live in other places, although maintaining the culture with differences. And so I think this is going to be very important for all of us, for science in general, because we're going to learn a lot about what happens, what is the contribution from environment to cancers, which is something that we suspect is important, but we really don't have much of hard data. We know that certain environmental exposures in certain countries cause certain things, but we don't understand, how is that colored with the ethnic background, with the personal background that we carry. And so I think this is really a great opportunity, and I commend you all for moving as fast as you have.
Yep. Thank you very much for your kind words and the encouragement, and for the continued support from all of you who are here, and many people
you cannot get us away, even if you try. If you tell us don't come ever, we don't want to see you, I'm sorry, we're still knocking on your door.
We haven't even thought about in the dreams either. So sounds good. Yeah, I think my time is over, and I'm going to hand into Disha again. Please, and thank you all again.
Thank you.
Thank you.
Thank you for having us.
Thank you. Thank you, Dr. Radhakrishnan. Before we move ahead, I'm going to ask all of you to, most of you already have your camera on. We're going to end this session with, of course, a picture. How can we not do that? So requesting the others to please switch on their camera so we can take not a picture, but a screenshot for sure.
This is the equivalent of a group picture
yes.
in the virtual age.
Yes.
Ankita, you are not on the camera. Yeah,
hey, here they are.
Nitya, are you gonna?
Nitya, I think.
I'm sorry, I don't want to single someone out, but it'd be nice if we get everybody on the camera.
And Parthi and the control room, I think. Please show yourself. Okay, who is taking the picture?
My ugly mug looks better. No, it does not.
And who is taking a picture, by the way?
I think we have multiple people. Part 3 also, I'm also taking it. So
okay, great. So I think this was day 1. We still have day 2 and day 3. So looking forward to it. And thanks as always to everyone.
yes,
Thank you for inviting us.
before we move on to the vote of thanks by Dr. Anamika, I'm just going to remind everybody of the sessions tomorrow, starting, the conference that starts tomorrow. Just a few housekeeping tips. All the sessions will be on the same website. Participants will log in on the same portal. If you haven't, or if you have somebody who will be registering, it's the same process: register, verify your email, log in, enroll, and join. So everybody will get a reminder, if not today, just before the session tomorrow for sure. So please check your emails, make sure you receive it. It's a reminder, but I think you all are all set for the next 2 days. In case of any technical issues that you encounter during the sessions, or during login, just send us an email, ctcr.tcga@gmail.com. We are responding to them immediately. The session tomorrow will start at 2 p.m. Indian Standard Time, and it will end by 10 o'clock at night, Indian Standard Time. Agenda is available on the website. The inauguration of the conference is between 4 to 5 p.m. IST, and this is going to be done by our Honorable Minister of Science and Technology, Government of India, Dr. Harshvardhan ji. He is the chief guest. And we also have Dr. Shekhar Mande and Dr. Zenklusen, who will be our chief guests. So we definitely look forward to seeing you all then. Make use of these sessions, and let's all be on time. That's all from me. Thank you, and over to Dr. Anamika.
With this question and answer session, our pre-conference workshop ends here. We are truly grateful to all our faculty members who have joined us today from all across the globe for their time and active participation. Thank you, Dr. JC, Dr. Wong, Dr. Anna, Siriak, Dr. Go, Katie, Benjamin, Kyle, Dr. Tan, Dr. Nitya, and Dr. Anguraj for very insightful sessions. I'm sure our participants have learned more today on cancer multiomics, latest breakthroughs, and the progressive way forward for clinical research. I would also like to extend my sincere gratitude to all our organizers, the Cancer Genome Atlas, Prashanti Cancer Care Mission, Indian Institute of Science, Education and Research, Pune, Institute of Genomics and Integrative Biology, New Delhi, Institute of Cancer Research, UK, DBT Wellcome Trust India Alliance, and Persistent Systems, and other co-organizers, endorsers, and partners for their constant support, without which this event wouldn't have been possible. An event like this cannot happen overnight. It requires precise planning and keen eye for details. We have been fortunate enough to be backed by a team of highly enthusiastic, self-driven, and dedicated professionals. Thanks to all the organizing committee members, Santosh, Srikant, Disha, and our volunteers, Devki, Sachin, Ashwini, Sanket, Siddharth, Ankita, and Persistent Systems marketing team, and many other members behind the scene who were a strong support system for this event. Thanks to both our moderators, Sabri and Disha, for making live question and answer session engaging and insightful. I would also like to thank our educational supporters, Illumina, 10x Genomics, and Premas Life Sciences for their support. We are also grateful to admin team members of Persistent, PCCM, and ISA Pune for logistic arrangements. I would like to take this opportunity to place on record heartfelt thanks to our technology partner, Sachin, and his entire team, Alok, Parthvi, Clarion, Ishika, for their continuous 24/7 support to execute this digital workshop seamlessly, and hassle-free. Last but not the least, thank you all, the participants, for your enthusiastic participation, and see you tomorrow for some more exciting sessions. Thank you.
So thank you, everyone. I will be in touch, and there are 2 more very interesting and busy days ahead of us. So with that,
I have a question for you, Santosh, before you close in. Did I get right that we use the same link that we used today for tomorrow's session?
No, you will receive in the next half an hour another link.
Another link. Okay. Okay.
Yeah.
Good.
So there will be a faculty room that we are creating, which will be completely different than today. So please await my email on this.
Yeah, I'll keep an eye open.
Sure.
Well, shouldn't you be going to sleep right now?
It's only 10 p.m. in India.
Only
yeah, right.
Only
In
10.
India, nobody goes to sleep before midnight.
He doesn't sleep. He's always awake.
Oh, you're right. He does not sleep. I forgot that.
Non-stop.
Yeah, he's on email 24/7.
All right. I hope next year we will have a big party in Pune or in India. Let's see, wherever it is.
Oh, well, that would be lovely to have. Without masks, being able to shake hands, That would be. Hey, Anamika, how are you?
I'm good. Thanks, Jesse.
Look at her. She's there.
Wow. Thanks, Katie. Awesome. I think this is bringing back 2019. So.
Yes, that's exactly it. The band is back together.
Great.
Great to see you all.
Great.
All right.
Thank you, everyone.
Great. Thank you all.
Thanks.
Take care. Stay
Bye-bye.
safe.
Bye.
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 Nice.